Hello dear students. In the previous class we were discussing about the volumetric analysis. Okay, so in the volumetric analysis again the classification. So mainly 5 types: acid base titration, non-aqueous titration, precipitation reaction titrations, redox titration, and also complexometric titration. So not only that, even the depending upon the condition, the acidometry titration or the alkalimetry titration. So here the acidometry titration nothing but the process in which it involves the quantitative analysis of the base, nothing but alkali, with the use of a standard solution of an acid. Okay, so in brief we will use the acid in the acidometric titration. So in alkalimetry, the process which involves the quantitative analysis of an acid with the help of a standard solution of a base or alkali is termed as mainly alkalimetry. Okay, so in the first one, the acid base titration. So acid base titration is a type of volumetric analysis which involves the neutralization reactions between the acids and base, and where the salt formation will be there with water liberation. So in this reaction, acid is nothing but it is a substance. When it dissolved in a water, it will undergo further dissociation and forms the H plus ions. And just similar to that, only base is a substance. When it dissolved it in a water, it gives the hydroxyl ions. So OH minus ions will get liberated. Okay, so in this commonly we will use the acids in this type of the titrations are hydrochloric acid, even the sulfuric acid. So alkali nothing but mainly sodium hydroxide, potassium hydroxide, and sometimes sodium carbonate also. So in this the indicators are used. Okay, so acid base indicators. Okay, so these are helpful for the detection of endpoint in these titrations. Okay, so it will be used in a very small quantities. These indicators are generally organic dyes which show the different colors in the dissociation and undissociation forms. So this color change for the indicators is mainly based on the pH of the reaction solution. So commonly using the acid base indicators or phenolphthalein indicator, methyl orange, methyl red, and also the phenyl red are the very different indicators which are used in practice. Okay, so in this the some examples we can study. So here the sodium bicarbonate assay and boric acid assay, and sometimes ammoniated mercury, and another one is the ammonium chloride assay. So all these assays are mainly based upon the acid base reactions, nothing but titrations itself only. The sodium bicarbonate, which commonly we will call it as a baking powder, NaHCO3. So this sodium bicarbonate is based on the acidometry. So as I already told, acidometry titration is nothing but the process which involves the quantitative analysis of the base with the use of the standard solution of an acid. Okay, so in this the sodium bicarbonate, the titrate will be sodium bicarbonate. Okay, so that one we will take in a conical flask, and titrant is 0.5 normality sulfuric acid. The indicator will be methyl orange, and end point the appearance of the yellow color will be there. Okay, so here the methyl orange indicator will be used, where the pinkish red will get converted to the yellow color. So that will be the end point in this titration. Okay, the sodium bicarbonate is assayed by acid base titration, especially acidometry method. The sodium bicarbonate is titrated with the sulfuric acid or the hydrochloric acid, preferably sulfuric acid, that is also 0.5 normality concentration. Using methyl orange as an indicator, the endpoint is the color change from the pinkish red to yellow color. So in this two molecule of the sodium bicarbonate NaHCO3 react with the sulfuric acid, which gives the sodium sulfate, and carbon dioxide will be get liberated with 2 molecule of water also get liberated. So reaction is also very important. Then related to the procedure. So in this process, the 1 gram is weighed accurately sodium bicarbonate, and it is dissolved in 20 ml of the water. Then it is titrated with the 0.5 normality sulfuric acid using the methyl orange as an indicator. So here one is each ml of the 0.5 normality sulfuric acid is equivalent to that of the 0.042 gram of the sodium bicarbonate. So this is the sodium bicarbonate assay. The another important acid base titration example is assay of boric acid. So in this titrate is boric acid, okay, which one we will take in a conical flask, and titrant 1 normality sodium hydroxide, we have to take that in a burette. Indicator is phenolphthalein and endpoint is pale pink color. So it may be analyzed by the titrometric method. So as boric acid is a very weak acid, it can't be titrated accurately with the standard alkali, and here when it is dissolved in a glycerin water mixture, it acts like a strong monobasic acid. Okay, so boroglycerate formation will be there and can be titrated with the standard alkali using the phenolphthalein as an indicator. So you can notice the reaction here. The glycerol react with the boric acid base, there is a formation of the glyceroboric acid. Okay, so here the procedure, you have to dissolve the accurate quantity, weight quantity of about 2 gram in a mixture of 50 ml of water and 100 ml of glycerin, which is previously neutralized to phenolphthalein. Then now titrate with the 1 normality sodium hydroxide using phenolphthalein as an indicator. So finally endpoint colorless to pale pink formation will be there, so that indicates the chemical acid base reaction completed. Okay, so here the each ml of the 1 normality sodium hydroxide is equivalent to that of that here 0.06183 gram of this boric acid H3BO3. Okay, so another important compound that is here the ammoniated mercury, so which is also called as amino chloride of mercury. So it is based upon acid base titration. The sample is dissolved in the solution of the potassium iodide first, then the precipitate of the HgI2 mercuric iodide dissolves in the excess of potassium iodide with the liberation of the equivalent amount of alkalis as ammonia and also potassium hydroxide. Okay, so which are titrated against the standard hydrochloric acid using here the indicator methyl orange. Okay, again same thing, at the end the yellow color formation will be there, so that indicates endpoint. Okay, so here the procedure you can notice, weigh accurately about 0.25 gram and transfer to a spotted flask. Okay, stoppered flask containing 50 ml water and 3 gram of potassium iodide, and shaking occasionally till it dissolves. The liberated alkali is titrated against 0.1 and hydrochloric acid, and in this the indicator is use methyl orange. Then the another acid base titration, the important example, that is ammonium chloride. Okay, so in this assay again same here the acid base titration reaction will be takes place. So here the ammonium chloride titrate is ammonium chloride, titrant is sodium hydroxide, that is 0.1 normality sodium hydroxide you have to use in this ammonium chloride assay. Okay, and indicator is phenolphthalein and endpoint will be pale pink color. So this assay is based on the acid base titration, and in which the specified weight of the substance is dissolved in water and treated with formaldehyde solution, liberating the equivalent amount of this hydrochloric acid. This hydrochloric acid is determined by titrating with the standard sodium hydroxide by using the phenolphthalein as an indicator. Okay, so you can notice the reaction here. The four molecules of ammonium chloride react with the 6 molecules of the formaldehyde, which forms the hexamine, and also 4 molecules of hydrochloric acid. Okay, so those 4 molecules of the hydrochloric acid react with the sodium hydroxide after the titration reaction, under titration reaction. Okay, so in this the procedure, you have to weigh accurately about 0.1 gram of sample and dissolve that in a 20 ml of water and add a mixture of 5 ml of the formaldehyde solution, which is already previously neutralized, to dilute phenolphthalein solution, and 20 ml of the water. Allow the reaction to take place for 2 minutes and titrate slowly with 0.1 normality sodium hydroxide by using the solution of the phenolphthalein as an indicator. So in this, the each ml of 0.1 normality sodium hydroxide is equivalent to that 0.005349 gram of ammonium chloride. So all these assays based on the acid base titrations itself only. So assay of sodium bicarbonate, assay of boric acid, assay of ammoniated mercury, nothing but amino chloride of mercury, and the last one important thing, that is assay of ammonium chloride. So all these are based on the acid base titrations.